My name is Stephen Shuster, and I'd like to talk to you today about using common garden data to simulate selection and evolution at the community level. In 1970, Richard Lewontin made the following statement. He said, Darwin's scheme embodies three principles: phenotypic variation, differential fitness, fitness is heritable. When they hold, a population of any kind, individuals or higher levels, will undergo evolutionary change. With that in mind, I'd like to talk about the distinctive patterns that characterize ecological communities that are associated with foundation species. In terrestrial and aquatic environments, foundation species are species that exert a disproportionate influence on their surrounding communities, either by creating stable environments or imposing selection in particular ways. Community genetics characterizes ecological communities that are associated with foundation species, and it does so using several tools. The first tool is common gardens. Common gardens, in the context of community genetics, is a way to take a look at the relative influences of environment and genotype on community organization. In this particular case, we're looking at cottonwood communities planted in a common garden in Utah. And the goal of this kind of experiment was to vegetatively propagate individual cottonwood trees and examine the relationship between those genotypes and the kinds of species that become associated with them. A second tool is a multivariate statistical approach known as non-metric multidimensional scaling, or NMDS. NMDS, when used to study community organization, captures the phenotypic outcome of trait interactions among host and symbiont species, in which each community represents a single NMDS score. Using this approach, common and rare symbiont species are treated equally by NMDS, and so the observed score values really do represent community-wide patterns. And for the purposes of this experiment, each NMDS score identifies a community phenotype for each clonal replicate within each tree genotype. But, like a quantitative trait, it provides no specific information on the genetic or the ecological basis for that score. Over the past 30 years, there have been literally hundreds of studies that have been conducted to examine the effect of genetic variation on population and community traits that are important in community evolution. And the overriding theme for these studies is that phenotypic covariance exists among communities that are located on genetically related host organisms. The inference from these studies is that interspecific effects imposed by hosts on their symbionts are heritable traits, or what we like to call in the cottonwood ecology group, interspecific indirect genetic effects, or IIGEs. Now, the majority of these studies, phenotypic covariance of symbiont communities has been documented using broad sense community heritability. And broad sense community heritability measures the phenotypic covariance of symbiont communities unrelated to cottonwood trees, or for that matter, any host organism that you're studying. Looking at the total variation in community phenotype, when groups of host organisms are clones, as they are in the cottonwood studies, broad sense community heritability equals the proportion of the total phenotypic variation that can be explained by host genotype. In other words, broad sense community heritability describes the contribution of all genetic factors influencing community phenotypic variation. Now, the importance of community genetics is especially true in the Anthropocene, because as we all know, climate change is imposing community-level selection on a wide scale. If individual trees have communities of organisms associated with them that may consist of hundreds of species, when individual trees or groups of trees or entire ecosystems of trees are extirpated, it's pretty clear that there are evolutionary consequences for biodiversity and conservation. This is a talk that Tom Whittam gave earlier in this meeting, and he made this comment as well. So there are three questions I'd like to address today. The first, is it possible to simulate community-level selection in common gardens? Secondly, what parameters exist for documenting evolution at the community level? And finally, what do these parameters mean? The first question can be answered using a study that was conducted by Art Keith in 2010, where he documented the repeatability of cottonwood communities on clonal replicates. in a study that was in a common garden data was collected in between 2004 and 2006. What we're looking at over here are the individual genotypes of trees that are color-coded, and the crosses here represent the NMDS scores that comprise these communities. What Keith showed was that community phenotypes across these 3 years are stable. They're genetically based interactions between trees and arthropods. The repeatability across these 3 years was over 92%. And the broad sense community heritability in each of these years ranged from 0.6 to 0.68. There was no temporal autocorrelation explaining these results. And so this was an interesting result because it showed that communities are stable from 1 year to the next. And this is particularly interesting because given that these communities were in northern Utah, each year leaves fall off of the trees and the communities of arthropods become reestablished on those trees. Now, what we did was we took those NMDS scores, and this is a summary of what the data look like. The NMDS scores come from the abundances of 102 arthropod species across 44 genotypes of trees. And the color coding represents the different clonal replicates of these trees. Distilling these arthropod abundances down to a single NMDS score for each tree genotype gives us a way to impose selection on these trees in this common garden. If you look closely, you'll see that the NMDS scores range from negative to positive values. We took the largest negative values and the largest positive values, used these values of NMDS to identify our individuals in the 2005 population, and then took the highest and lowest scores of these values. We then identified our 2006 scores, conducted a new ordination, and essentially produced 2 episodes of selection. The 2 episodes of selection in 2004 and 2005 showed that even though all of the values were close to 0, by 2005, our community phenotype had diverged away from 0. And by 2006, our NMDS scores were significantly different from our initial values. When we randomly selected communities, we found no significant pattern of NMDS scores in 10 replicates. So the answer to the first question is it possible to simulate community level selection in common gardens? The answer is yes. The next question is, what parameters do we use for documenting evolution at the community level? Well, you may notice that we've considered situations where we're documenting a multivariate value for a community phenotype. And so we can use very similar statistics that are used in quantitative genetics. Here, the red line represents our community phenotype distribution before selection. The hatched area over here represents our community phenotypes that were part of our selected group. And so our community selection differential, S sub C, is equal to the average community phenotype of the selected population minus the average community phenotype before selection. Very much like the breeder's equation. The response to community level selection is equal to R sub C, and that response to community level selection equals the average community phenotype in the year following selection minus the average community phenotype before selection is imposed. So we now have values for our community community selection differential, as well as our response to community level selection. And we can now calculate a realized community heritability. The realized community heritability is the ratio of the response to selection to the selection differential, just like the breeder's equation, except in this particular case, because we're imposing community level selection, we have a breeder's equation for community genetics. The values that we got for our realized heritabilities are similar to those that would be achieved for imposing selection on quantitative traits at this level and at this intensity of selection. And interestingly enough, our values for broad sense community heritability had lower confidence limits that overlapped with our estimates for realized heritability, suggesting that broad sense community heritability and realized community heritability are similar in their magnitude. So, the parameters that are used for documenting community evolution are very similar to those for quantitative genetics, but let's see what those parameters mean. Community phenotype we can identify as the richness and abundance of individual species on individual host genotypes as a result of the genetic interactions that exist among community members, or IIGEs. Communities can be quantified using non-metric multidimensional scaling, and therefore it is possible to identify a specific community phenotype associated with individual host organisms. Broad sense community heritability summarizes the phenotypic covariance of symbiont communities associated with particular host genotypes. And it also identifies the evolutionary outcome of selection that's operating within a community context. Community level selection is the outcome of selection operating at multiple levels, not only selection operating within a community context, but also as a result of community level selection. It can be quantified using metrics that are very similar to those used for quantitative traits, and it can lead to the differential survival and/or proliferation of communities, depending on whether it's a common garden environment or a natural community. Realized community heritability summarizes the phenotypic covariance of symbiont communities from one episode of selection to the next. And it also summarizes the evolutionary outcome of selection acting at the aggregate community phenotype. So we can define community evolution as a detectable change in the average community phenotype as a result of selection. And this change in phenotype will result from selection acting in a community context, as well as community level selection. And if you'd like to read more about this idea, you can consult not only Whittam et al. In the Annual Review of Ecology and Systematics, or Shuster et al. In Ecology and Evolution. In conclusion, broad sense community heritability summarizes the genetic interactions that underlie community phenotype. Broad sense community heritability also summarizes the phenotypic outcome of selection within a community context. Because genetic interactions are preserved with community level selection, this is the explanation for why broad sense community heritability and realized heritability might be equivalent. And so community heritability not only summarizes the phenotypic covariance among genetically related communities, it also predicts the evolutionary response to community level selection. And a large number of collaborators were responsible for making this research possible. Thanks for listening.